Hello students, welcome back to Edupedia word videos. My topic for the presentation is 6th section of the chapter Transport in Plants. In this section of the video, we will continue to discuss about the vascular tissue system. Students, I have already taught you that vascular tissue system is a unique characteristic that is found in vascular plants, which are also called as tracheophytes. So vascular plants and tracheophytes are same. Vascular tissue system comprised of vascular bundles, xylem and phloem. Now question arises, what is xylem? We very well know that it is a conducting tissue which is present in plants. Students, when your body is thirsty and hungry, what do? You drink and eat, right? All plants, they need to be able to drink and eat too. Plant and trees might not have mouths, but they do have special ways that they can get their nutrients. So xylem and phloem are the name for the special pipe-like system that allows water, mineral and food to get delivered to all parts of a plant body. So xylem, it is a conduction tissue which is present in plants, and the xylem term was proposed by Nageli. He was a scientist. He proposed a term called xylem. Xylem, it is derived from a Greek word xylos, which means xylem main function is conduction of water and minerals from root to leaves. An additional function is to provide mechanical support, such as secondary xylem. And two types of xylem in plants are present. Which are those? First is primary xylem, which is formed from promeristem during primary growth. Students, I have already taught you about meristem, promeristem in the chapter Anatomy of the Flowering Plants. You can refer to that for better understanding. Okay. So two types of xylem in plants are found. First is primary xylem and second is secondary xylem. Okay. Primary xylem, it is formed from promeristem during primary growth, whereas secondary xylem, it is formed from vascular cambium during secondary growth. Secondary xylem, it increases the girth of a plant body because it involves in secondary growth of a plant body. Okay. Primary xylem versus secondary xylem. Primary xylem, xylem in the primary plant body is called as primary xylem, and it is developed from procambium. It is organized as bundle along with phloem. That means xylem and phloem, they are joined together with cambium, and that acts as an entity. Okay, and the main function of primary xylem is the conduction of water and minerals. Then comes secondary xylem. Secondary xylem, it is present in secondary plant body. Okay, and it is developed from vascular cambium. To be more precise, lateral meristem, and the main function of secondary xylem is conduction of water and mineral. An additional function of secondary xylem is to provide mechanical support, since it increases the girth of a plant body and thus it provides mechanical support. Okay. As you can see that this is secondary phloem, which is represented by this dark yellow color. This is primary phloem, which is derived or developed from procambium. This is primary xylem. This one is primary xylem. Okay, and this is secondary xylem, which is represented by this blue color. This is vascular cambium. This is pith, which is a central part. And this is cortex, which is a ground tissue. Okay. Let's see the zoom view of it. This is primary xylem. This is new secondary xylem. This is dividing vascular cambium. And this is new secondary phloem and primary phloem. Okay. Now we'll talk about the components of xylem. Xylem, it is composed of several types of cells. Some cells are living and some are dead. Okay, and the elements of xylem are 4 types: tracheeds, vessels, fibres and parenchyma. Okay, So xylem, you can say that it is a complex permanent tissue which is specialized for the conduction of water and mineral substances in a plant body. And xylem is a heterogeneous tissue which is made up of 4 different type of cellular elements, which are those trachedes, vessels, fibres and parenchyma. Okay, and some cells of the xylem are living and some are dead. Now we'll talk about the trachedes. The trachedes and the trachea, they are described as essential elements since they are directly involved in translocation of water and mineral substances. And it is a fundamental cell type in xylem. Trachedes, they are elongated tube-like. They are non-living at maturity, and mature cells is empty without protoplast. That means in trachedes, no protoplast is there. Cell with secondary cell wall. And secondary cell wall is highly lignified. That means it has a deposition of lignin. Cells of trachedes are polygonal in shape, and the average length of trachedes is 5 to 6 millimeter, and both the ends of a trachedes are tapering. Okay, and phylogenetically, if you see, then trachedes are the most primitive type of cell found in xylem. Okay, so this is the vessel member, this is the trachea, which is also called as vessel. This is tracheid. Tracheid is a fundamental cell type which is found in xylem, and they are elongated tube-like, as you can see that it is just like a tube. They are non-living at maturity. That means initially they are living, but as they mature, they get converted into non-living. And mature cell is empty without protoplast, and cell with secondary cell wall is highly lignified, and cells of tracheid, they are angular and polygonal in shape. Tracheeds, end of the tracheeds of a secondary xylem are kysel-like. See, it is like a kysel. Okay, so this is the end of a second xylem, or you can say the end of a tracheid of a secondary xylem is just like a kysel. Okay, and major part of a cell is imperforate. That means the major part of a tracheid is imperforate. That means it does not have pores, whereas it has pit pairs. See, this is the pit. Okay, whereas it has pit pairs at a common wall. See, this is the common wall of this and this. This is the common wall. So it has one pit pairs, and pit may be circular or bordered pits. Tracheeds are the only xylem element which is found in pteridophytes, and in angiosperms, tracheid occurs with other xylem elements. Okay, and in primitive angiosperms such as thymus, trichodendron, tetracentron, xylem is only composed of tracheeds, but no vessel is found. Okay, vessel is completely absent in it. So this is in the primitive angiosperms such as thymus, trichodendron and tetracentron. Tracheeds. Now we'll talk about the pattern of secondary thickening in tracheeds. Students, as we know that secondary cell wall material, they are laid down on the lateral wall of the tracheeds, and many patterns of secondary thickening occur in tracheeds, and some of them are annular, spiral, reticulate, scalariform and pitted, right? So trachea type by cell wall thickening are annular, spiral and reticulate. Now we'll talk about the tracheid thickenings. In this we'll study the longitudinal section of a xylem, that will give us a better understanding. See, these rings are deposition of lignin. This is showing the deposition of lignin in the secondary wall. Okay. As a result, a xylem vessel, it exhibit different type of secondary thickening. On the basis, xylem vessel can be distinguished into 5 types. So first is annular. Annular, you can say that in which secondary thickening is in the form of ring, which is placed more or less at equal distance from each other. As you can see that these are the lignin rings which are placed more or less at equal distance from each other. Okay, so this is annular thickening. Now we'll talk about the spiral thickening, wherein lignin rings are in the form of spring or helix. So spiral trachieds are those in which secondary thickenings are present in the form of helix or coil, or you can say just like a DNA. Then comes network like, which is called as reticulate. Reticulate trachieds are those in which secondary thickenings are irregular and they appear in the form of network, or it forms a mesh like network. So that's why it is called as reticulate thickening of tracheid. Then comes ladder like, which is also called as scalariform tracheid, in which secondary thickenings they appear in the form of cross bands. As you can see, it appears in the form of cross bands, and that resembles the step of a ladder or a staircase. Okay, then comes pitted trachea, in which secondary thickening result in the formation of depression on the primary wall, which is called as pith. Now we'll talk about the annular thickening in detail. The deposition of lignin takes place in the form of ring on the inner surface of a cell wall, as I have just shown you in the diagram. See, this is just like it. Okay, so this is annular and this is a spiral thing. These lignified rings, which are placed one above the other like coins, they leave sufficient space in between each other. As you can see that they are placed in more or less at equal distance from each other, right? And the gap of wall remain unthickened. Such thickenings, they are commonly found in vessels and trachees. Okay, now comes spiral thickening. In such cases, the deposition of thickening material, that is lignin, takes place in the form of complete spiral band. See, it is like a spring in which secondary thickening are present in the form of helix or coil or like a spring, and the number of such bands may be one or more than one, as you can see. And this type of thickening is commonly found in vessels or trachea of angiosperms. Okay, now we'll talk about another thickening, and that is scalariform thickening of tracheeds. In such thickening of cell wall, the lignin is being deposited in the form of transverse rod like this, and thus it is called as scalariform or ladder-like, and the unthickened areas between successive thickening layer appear as elongated transverse pit. As you can see, it has formed a transverse pit, and this type of thickening are common in xylem vessel and tracheeds of Protoxylem. Okay, then comes fourth type of a thickening, which is called as reticulate thickening, and it is also called as net-like thickening. It is a net-like distribution of secondary material, just like this. It forms a network-like, and in which secondary thickening are irregular and it appear in the form of network. So this is scalariform thickening and this is reticulate thickening. Now we'll talk about fifth thickening, which is called as pitted thickening. Secondary wall material is evenly distributed and cell wall more or less is of uniform thickness. Many pit, they are distributed over entire wall like this. As you can see that many pits, see these small are the pits, and many pits are distributed over entire wall, and most advanced type of thickening is called as pitted thickening. And pits may be circular or elongated bordered. So this is pitted thickening. Okay, now we'll talk about the tracheeds structure in relation to function. Tracheeds, they are especially adapted for water and mineral conduction. This is the fundamental cell type of xylem, right? And cells, they are elongated with tapering ends. They are devoid of any protoplast, because when it matures, it get converted into non-living structure. Initially it is living, but as it progresses or as it matures, it gets devoid of protoplasm, and thus it gets converted into non-living structure. Tracheates, they are thick lignified secondary cell wall, and end walls, they are provided with pit pairs. Cells are placed end to end to the long axis of the organ in which they occur, and water and mineral passage, it takes place through pit membrane. Okay, so this is the bordered pit. As you can see, this is the middle lamella, which is represented by this blue color. This is primary cell wall, this is secondary cell wall, this is the pit area, pit. Okay, this is torus of pit. It acts as a wall for closing and opening of pit, as you can see. Okay, so this is the bordered pit which is present in the cell wall. So cells, they are placed end to end to the long axis of an organ in which they occur, and water and mineral passage, it takes place through pit membrane, and the torus of pit, it acts as walls. Okay, now we'll talk about another cell of xylem, and that is xylem vessel. Xylem vessel, it is also called as trachea. They are shorter cells which are shorter than tracheeds, okay, which we have just studied. Cells of vessel, they are non-living at maturity, and cells, they are devoid of protoplast at maturity. They are made up of tube-like cells, and usually they are absent in pteridophytes and gymnosperms. Please note that this is very important question that comes in pre-medical examination. Okay, so xylem vessel, it is usually absent in pteridophytes and gymnosperms. Vessels, they are absent in aquatic plants, they are absent in parasitic plants, and some succulents do not have vessels. And please note that in xylem vessel, cells, they are arranged as series in an end-to-end fashion. Cells, they are arranged parallel to the long axis of an organ, and numerous pits are present in the lateral wall for communication. And component of vessel, they are called as vessel elements. Okay, so we'll talk about the structure of a vessel, which is made up of series of cell which is placed end to end. It gives long tube-like appearance, as you can see in the diagram, and each cell is called vessel membrane. Okay, and vessel membrane have perforation, that means it has many pores at the end wall. Usually vessel membrane, they are shorter than trachees, and diameter is much more than trachees. So this is xylem vessel. Okay, this is the xylem vessel zoom view. This is the thickened band. This is the lumen of a vessel, and this is long vessel like, and this is the transverse section through dicot stem to show a xylem vessel. Okay, so dead plant cells, they are laid end to end without nucleus and cytoplasm with thickening cell walls, and that is xylem vessel. Okay, now opening or pores in vessel membrane wall is known as perforations. Okay, opening of pores in vessel membrane wall is known as perforations, and perforation plate is the region of a vessel with perforation occurs. See, this is the perforation plate from where perforation occurs. Perforation means that allows the passage. Okay, so usually perforation occurs at the end wall. See, this is the end wall, and here perforation plate is there, and usually perforation occurs at the end wall. Sometimes lateral perforation also occurs. That means this is the lateral part of it. So sometimes it is seen that perforation also occurs from this lateral side. Different type of perforation plates are seen. First is simple perforation plate. It is a plate with single perforation. That means only one pore is there that allows perforation, or that allows passage. Okay, to move in or out, right? And then comes multiple perforation plate, which has a plate with many perforations. Then comes scalariform perforation plate, wherein perforations, they are arranged in parallel series. Then comes another type of a perforation plate, and that is perforation bar, which is a wall region of a pore in scalariform perforation plate. Then comes reticulate perforation plate, wherein pores are arranged in reticulate fashion or in the mesh-like network. Okay, and then comes forminate type perforation, wherein many pores, they are arranged more or less circular pattern, and it is called as epiphysoid perforation plate. Okay, now we'll talk about the secondary cell wall thickening in case of xylem vessel. So secondary thickening of vessel is very much similar to the tracheeds thickening as we just studied. So there are 5 types of thickenings: annular, spiral, reticulate, scalariform, pitted. Pitted thickening is characteristic of metaxylem and secondary xylem. Okay, see, this is annular, this is spiral, as I have taught you in case of tracheed secondary wall thickening, and this is ladder like, which is called as scalariform. This is network like, which is called as reticulate, and this is a small pore like, which is called as pitted. Okay, pit on xylem, they are commonly bordered, and pit distribution pattern varies. So there are 3 basic patterns seen in which pits are distributed. So first is scalariform pitting, wherein elongated with pits in ladder like arrangement. Then comes opposite pitting, that means pit are arranged in horizontal rows in pairs. In pairs, opposite pitting takes place. And third basic pattern is alternate pitting, wherein pits or pores, they are arranged in diagonal rows. Okay, so 3 basic patterns of pit are distributed in. Now the third cell of xylem is xylem fiber, which is also called as xylary fiber. They are represented by dead sclerenchyma fibers that are found in between vessels and tracheeds. I repeat, xylem fibers, they are represented by dead sclerenchyma fiber, and they are found in between vessels and tracheeds. Xylem fibers, they are meant for providing mechanical support to the essential elements. Okay, it has no protoplast at maturity, hence they are considered as dead cells. Mainly, these fibers, they provide strength to the vessels. They are present more abundantly in secondary xylem. Okay, so this is xylem fiber, this is septate, and this is aseptate. Students, it is seen that there are 2 types of xylary fiber are found. First is fiber tracheed, and second is libriform fiber. Okay, 2 types of xylary fibers are seen. Xylary fibers are longer than tracheeds. Please note that. Okay, it is very important to know that xylary fiber, they are longer than tracheeds. And fiber tracheeds, they have less developed bordered pits, and libriform fiber have small pits. Gelatinous fiber, they are found in tension wood, is also a xylem fiber. Now we'll talk about the gelatinous fiber, although it is not there in NCERT book, but for knowledge sake I'm explaining you gelatinous fiber, though it is not very important from your course point of view. Secondary cell wall do not have lignin but have cellulose. This part of cell wall appears as gelatinous, and gelatinous fiber, they are highly hydroscopic. That means it absorbs water. Okay. Then fourth type of cell which is found in xylem is xylem parenchyma. It is a component of xylem. It is living cell, cells with cytoplasm and prominent nucleus. Okay, that means xylem parenchyma has so much of cytoplasm and prominent nucleus. Thin cellulosic wall is there. Okay, and it has lignin deposition in secondary cell wall, and where very rarely parenchyma in secondary xylem undergoes secondary growth. Parenchyma store starch, oil and other ergastic substances. Students, it is seen that two type of xylem parenchyma is there. First is axial parenchyma and second is ray parenchyma. So this is axial parenchyma and this is ray parenchyma. Okay, let's see what it is. Axial parenchyma, it is originated from elongated fusiform cambial cells. Okay, and it is arranged parallel to the long axis of organ in which they occur. As you can see that this is the long axis of a organ, so it is arranged to the long axis of the organ in which they occur. Then comes another type of xylem parenchyma, and that is ray parenchyma. It is originated from ray initials of cambium. Cambium is found between xylem and phloem. Okay, and there are two types of ray parenchyma. First is procumbent ray cells and second is upright ray cells. So what is procumbent ray cells? It is a long axis of a cell that are radially elongated, and upright ray cells are the long axis of the cells which are vertically elongated. Okay, now we'll talk about the tyloses, students. Ray parenchyma and axial parenchyma, they develop protrusion. Okay, and these protrusion, they enter or invade trachedes or vessels. Invasion occurs when trachedes or vessel become inactive or damaged. Okay, then only invasion takes place. Tyloses, you can say that it is an outgrowth of parenchyma to the lumen of tracheid or vessels. Tyloses development occurs through the pit pairs. Pit pairs, as I've shown you in the diagram of tracheid. Okay, so tyloses development occurs through pit pairs connecting parenchyma and tracheids or vessels, and sometimes many tyloses originate, and tyloses completely fill the lumen and thus it blocks the path. So it is like very harmful for the conduction of water and mineral because it blocks the path. Okay, because of the protrusion or invasion that occurs when tracheid or vessel become inactive or damaged. Okay, so tyloses somewhat look like this, balloon like. Okay, so tyloses, nucleus and protoplasm of parenchyma, they are retained in tyloses. Usually tyloses stay as thin walled, very thin walled. Okay, as you can see that it is very thin walled. Sometimes secondary thickening occurs in tyloses, and tyloses significance is that it prevents the leakage of water from damaged xylem. Okay, so that's the significance of tyloses. But tyloses formation is harmful because it blocks the path. Okay, or you can say that it hinders the conduction of water and mineral. Tyloses, it accumulates resin, that's another significance, and it also stores or accumulates secondary material. They are responsible for characteristic odor of wood. Tyloses, it also prevents the degradation of wood by termites and mites. Okay, tyloses, it lowers the permeability of the wood. Tyloses, they are formed during normal heartwood formation or after an injury. Okay, so tyloses only formed when during normal heartwood formation or after an injury. I have already taught you heartwood formation in the chapter Anatomy of Flowering Plants. You can refer to that for better understanding. Okay, now we'll talk about the classification of xylem based on origin. Xylem is classified into two groups, which are those: first is primary xylem and second is secondary xylem. And this primary xylem is divided into two: protoxylem and metaxylem. As you can see that this is the stem transverse section, this is pit, These are the vascular bundles which are arranged in ring form. This is primary xylem, this is secondary xylem, this is vascular cambium, this is secondary phloem, this is primary phloem, this is cortex, which is also called as ground tissue, this is cork cambium, and this is periderm. Okay. So primary xylem. Xylem is formed from during primary plant growth. It is derived from Procambium. It consists of two parts, which are those protoxylem and metaxylem. So this is metaxylem and this smaller ones are protoxylem. The bigger ones are metaxylem and smaller ones are protoxylem. Okay. Now we'll talk about the protoxylem. It is a first form xylem. Then later it forms metaxylem. Differentiate and mature well before the organ complete its elongation. Okay, when subjected to high stress due to organ development, usually protoxylem gets destroyed. Okay, when it is subjected to high stress due to organ development in plant body, usually protoplast or protoxylem gets destroyed. Protoxylem persists in root, then stem. In monocot students, protoxylem elements, they collapse and they form channel, which is called as protoxylem lacunae. Okay, it forms a lacunae like structure which is channel. Okay, so in monocot, protoxylem elements, they collapse and they form channel like, which is called as protoxylem lacunae. Protoxylem lacunae, they are surrounded by parenchymatous cell, and protoxylem lacunae, they are not formed in dicot. So it is a unique characteristic which is found only in monocots. Okay, protoxylem contain less tracheary elements and more parenchyma. Okay, and protoxylem tracheeds, they are narrow. They have annular or helical secondary thickening. See, this is the cortex, this is the endodermis, this is protoxylem, this is metaxylem. Now we'll talk about the metaxylem. It is a later formed primary xylem. It is formed from fascicular cambium and interfascicular cambium. These are not subject to much stress and strain, thus it gets retained. Whereas when protoxylem is subjected to much stress and strain due to organ development in plant body, it gets destroyed. But unlike protoxylem, metaxylem is retained even after stress and strain. Okay, and the secondary cell wall layer thickening is in the form of reticulate or pit. Cells are wider, as you can see that this is metaxylem and cells are wider. The xylem elements, they are larger in diameter. See, just compare this cell and this cell. This cell has very small diameter in comparison with this cell. Okay, so you can say that metaxylem, which is this, it has larger diameter, and plant without secondary thickening, metaxylem is functional throughout. So this was all about it. Stay tuned for more awesome video lectures. Thank you.